He's a researcher here at CCMAR in Bascal Lab. His research has been conducted in the field of osteotoxicity mostly, and he uses fish cell lines as an in vitro model for his studies. And is also a CCMAR partner in the recently established S2 Aqua Collaborative Laboratory. So Vincent, whenever you want.
Well, thanks, Gilles, for the introduction and for the invitation to give yet another seminar, CCMAR seminar. So today I would be talking about fish cell lines, and in particular about the work which is being done in the Bioscale lab at the CCMAR. First, I will give a brief overview and present some general information about fish cell culture. Then I will focus for a while on fish bone-derived cell line, which is a major research line in Bioscale lab. And finally, I will present some few slides about how the CCMAR is investing in the future regarding fish cell line. But before talking about fish culture, I would like to mention the free online resources that I have been using to collect data on fish cell line. The first one is the Celosaurus. It's a knowledge resource on cell line, which is available from the Swiss Institute of Bioinformatics. It catalogs or it lists about 125,000 cell lines and provide information on the cell type, on the species of origin, on the tissue of origin, etc., etc. And it contains information mostly on vertebrate cell line, also on insect cell line, but not on plant. And it's only about immortalized cell lines and stem cell lines. It does not talk or it does not present any data about primary cell culture. And I will give some few definitions about primary cell culture and cell lines in the next slide. I have also used the FishBase, which is well known from most of you. It's a database of fish species, and it provides information on taxonomy, geographical distribution, biometry, morphology, etc., and of course about habitat ecology. And finally, I have used the taxonomic information from the ETS database, especially to get reliable information about fish species. So before I start with the main presentation, I would like to give you just a clear definition of what is a cell line, because I will focus really on that today. So at the beginning, when you initiate a new culture of cells, you have a primary cell culture. And that primary cell culture contains or is composed of the first cells that came out of either the explant cell culture or the dissociation cell culture. So dissociation cell culture is usually for soft tissue where cells can be dissociated from the tissue using a gentle mechanical dissociation. On the contrary, explant cell culture is usually used for hard tissue, such as bone or cartilage, where cells cannot be dissociated from the tissue without damaging them, and when an enzymatic dissociation is needed to lose the cells. So cells of the primary cell culture will adapt to the in vitro cell culture condition, or they will die. At confluence, we will subculture them. And here again, either they adapt to the culture or they survive the cell culture, or they will die. And after some few passage, the cells that will survive and adapt well to the cell culture condition will form a cell line, okay? And that cell line can undergo senescence and eventually cell death, and we name this cell line finite cell lines, or they can get immortalized and acquire some capacity to divide indefinitely, and we call them continuous cell line. Okay, so I think it's important because I would mainly talk about cell line. I will not talk about primary cell culture. So now let's talk about the current situation. And actually, it's not. It's on December 2020. The situation about fish cell lines. So the first report of a successful culture of fish cell line date back from 1960. Okay, at that time, mammalian cell culture was a routine laboratory technique for quite some time. But the first cell line of fish origin was developed in 1962 by Wolf and Kimby from the rainbow-toothed gonad, and it's called the RTG2. And amazingly, 60 years after the development of this cell line, it's still in use, it's still working just fine. So today, in 2020, okay, some few months back, there is more than 700 fish cell lines that have been developed. Exactly 717, that's the number which is available, that's the number of cells which are available from the Celosaurus database. So they have been developed from a wide range of species and of tissue, and I will talk more about this in the next slides. So that number, it's kind of high if we compare with the other vertebrates, which are the amphibian and the reptilian, but it's much smaller if you compare to primate and especially human cells, which account for more than 92,000 cell lines and rodents. If we look at the data about fish cell lines, and specifically about the first record of the fish cell line. So the Celosaurus database have that information is the year of the first publication for a particular cell line. So if we look at this data, we can see that there is a steady increase in the last 6 decades. So the steady increase in the last 6 decades in the provision of fish cell lines. And we think that this is probably the consequence of an increased usage of fish in science, and which results from more research on fish species. And then here I would like to differentiate 2 different kinds of species. Those that are important for socioeconomic activities, such as fisheries and aquaculture, but also the fish species that are fish models, those that are important for biomedicine, okay? And then more research on fish resulted in more fish cell line developed, which is kind of normal because in vitro cell systems are necessary companion to in vivo systems, because they will provide insight into cellular mechanisms, into the signaling pathway that are important for biological process. We think also that this steady increase in the provision of fish cell lines has to do with ethical principle in science that in the last decades aim at a reduction in animal experimentation. And actually there is many research institutions and pharmaceutical companies that have already validated in vitro cell system as a suitable alternative to replace animal models. And here I give you just an example. It's that cell lines which is now used to replace the acute fish toxicity case in many cases, okay? So at this stage, I think it's important to mention that this fast provision of fish cell lines in the last decades largely benefited from the techniques, the reagent and the media that has been developed for mammalian cells. And when I talk about technique, it's the subculture, the cryopreservation or the transfection of the cells. About the reagent, it's the antibiotic, the antimycotics, or the basal medium that has been used for mammalian cells that have been successfully used for fish cells, okay? There is a special mention for the FBS, the bovine serum, which is used to grow fish cells. It may seem physiologically inappropriate to use bovine serum to grow fish cells, but actually it has been working quite well. And it also represents an enormous financial benefits, because it would be impossible to have enough fish serum in quantity sufficient to sustain fish growth worldwide. So it is economically very important that fish cells can grow using bovine serum. So we can say that the cultivation of fish cells and mammalian cells, it's basically the same. They use the same method, we use the same reagent, the same media. There is one thing that has to be adjusted, is the temperature of the incubation of the cells. So you all know that mammalian cells grow at 37 degrees, and then there is not that much latency. You cannot really decrease or increase by a few degrees. On the contrary, fish cells, which are cold-blooded animals, can grow in a wide range of temperature. And for cold water species, usually it's between 15 to 21 degrees. While for the warm water species, like the zebrafish, for example, which is a tropical fish, which will grow at 28 degrees, we can have a range of temperature from 22 to 33. Another reason to explain the fast provision of fish cell line is their capacity to spontaneously transform and acquire the ability to grow continuously. So nearly all the fish cell lines that are available from that Celosaurus had suffered that spontaneous transformation. That means that they were not induced for immortalization, for transformation, like most of the mammalian cells or from humans, from rodents. So fish cell lines can spontaneously transform towards immortalization. The reason, the mechanisms, we still don't know. Some people have proposed that it could be because fish cell lines are mostly aneuploid, or because they express a high telomerase activity. And I put here a small scheme for you to understand. So cellular senescence is associated with a reduction in the size of the telomere, and actually the telomere which are at the end of each of the chromosome. And actually the telomerase will kind of protect this telomere, will prevent them to reduce. So high telomerase activity could be the explanation why the fish spontaneously transform towards immortalization. It could be also the loss of the p53 protein, which is a tumor suppressor gene. But we still don't know. Anyhow, this... Anyhow, somebody speaking. Anyhow, this immortalization was a great advantage. It greatly improved the success rate of the development of fish cell lines. We have made we have seen that most of the cell lines from fish actually are developed from normal tissue, not from tumors. And that could be related to the fact that actually we do not need to develop a cell line from tumors, because they can spontaneously transform and get immortalized in culture. So let's analyze deeper the data that we found in the Celosaurus database. So I told you there is more than 700, exactly 770 cell lines available in that database. And it originated from 161 fish species that account for 62 family of fish. And 4 taxonomic group, and they are there. So the Neoteleostei, the Ostariophysi, the Accipanceriform, and the Protocamptopteridae account for most of the species, so 93%, and also for most of the cell lines, 97%, with a special mention to the Neoteleostei, which account for 47% of the cell line. It is important to note that not all the fish taxa, all the taxonomic group are represented with the cell lines. And one important group which is not there is the Holostean, and in particular the spotted gar. So the Holostean will be a group which is basically between the Chondrostei here and the Teleostei, okay? And these fish, especially the spotted gar, they have emerged as an important species for evo-devo studies. And some people think that it could be actually the bridge between human and teleostean biomedical models. Why? Because on the contrary to the teleostei that suffer a genome duplication event, or the chondrostei that suffer some various event of polyploidization, the holostean have a genome which is actually very similar to the one of the human, with no duplication of genes. So now if we use the data that we can find in the fish database about environment and the use of the fish that are the origin of these cell lines, we can see that freshwater and marine fish, there is slightly more cell lines from marine fish, but I would say it's kind of an equilibrium. It's 52% of versus 48. So it's kind of equilibrated. And if we look at the use of this fish at the origin of the cell lines by humans, you can see that most of them are in use for fisheries and also for aquaculture. Few of them in aquarium or even fewer in biomedicine. And actually this is according to the primary application of these fish cell lines, which has been the study of host-pathogen interaction to understand the disease outbreaks that you have in fisheries and in aquaculture. So a special mention for the biomedical model that represent only 7.5% of the fish species, but actually they represent 16.5% of the cell line available. So this is a number which is growing, and we have the goldfish, the zebrafish, and the medak as the major contributor of fish cell lines from biomedical model. So if we look a little bit about the tissue that are represented by these cell lines, the information in the Celosaurus was not sufficient, was not homogeneous, so I could take any information about the cell type. So the only information I could get was about the tissue at the origin of these cell lines. And the first thing that I could see is that most of these tissue are soft tissue. Okay, actually, only 12 of the 704 cell lines that were listed here were from hard tissue. Well, I will discuss a little bit later why this disequilibrium between hard tissue and soft tissue. Now, I will give you, I will not talk about all this tissue because it will be a lot of, it will be very boring for you, and it will be a lot of information that, that will take all the rest of the seminar. But I will just focus on the fin, which are very important because they are the one that with more cell lines. I will also focus on the embryo, on the gill and liver, and I will have a small word at the end for cancer cell type. So why the fin? Well, the fin is because probably we can say that fin is the anatomical structure which best define the fish, correct? And but then it's kind of hard to understand why fin cell culture was developed so frequently, and we think it has to be related to the easy access of that tissue. Okay, so we think that fish cell lines were developed to be species-specific and not organ-specific. In other words, the biologists wanted a cell line from a specific species, and what was the first organ easily accessible to prepare that cell line? It was from the fin. Okay, the cell lines from the embryo, it's slightly different. They are still used a lot for viral studies, but they were also developed to be used to test, to study the pluripotency of the embryonic stem cells. The gill and the liver cell lines are also very important for ecotoxicological studies. So the gills are organs that are directly exposed to the organic compounds or to the environmental pollutants, while the liver is an organ that plays a central role in xenobiotic metabolism. So these particular cell lines from gills and liver were particularly used in ecotoxicological studies. Now, a small word about cancer cell lines. This is a very atypic group, okay? They were among the first fish cells to be developed. And I think in the 90s, we had some cell lines from goldfish. But then since 2008, there is not one single cell line derived from cancer tissue, cancerous tissue, from tumor tissue. And then it's kind of difficult to understand. especially because fish, and in particular zebrafish, is becoming, there is a growing interest for fish and for zebrafish as human cancer model. So why there is no more cancer cells that are being developed? This is still puzzling, and we have no clue about that. So this slide, it's about a figure from a recently published paper from Tangarej et al. So it was published in 2021. And in that paper, they wanted to give an inventory. It's a world panorama of the contribution to the fish in vitro by country. So first of all, what is an in vitro? So this is in vitro. So this is a term that was coined by two persons from Canada, Niels Bolz and Lucy Lee, that are actually pioneers in the development and the usage of fish cell lines. And fish, an in vitro, actually, it's a catalog of cell lines. So for example, the truth in vitro will be the catalog of cell lines from truth. And here the fish in vitro is actually the catalog of cell lines from fish. And in that paper, they particularly focus on the fish in vitro from 2010 to 2020. And you can see here that the most important contributors to that fish in vitro are Asiatic countries, with China, India, South Korea, and Taiwan among the most contributor, the country that contributed most to fish cell lines. You have also Canada, and then you have here Portugal. And well, with 11 fish cell lines, it placed Portugal in the top 10 of the country worldwide to develop fish cell lines. And actually it's a leader in Europe. It's before Spain, Germany, France, UK. And well, this is our little moment of glory, because if you look into that paper, into the 11 cell lines that have been developed by Portugal, all of them were developed at the CCMAR by the Bioscale lab. Okay. So what about the availability of the fish cell lines? And actually the scenario is actually not that good. Okay. If you look into the Celosaurus database, only 5% of the fish cell lines are available in cell bank, such as the European Cell Culture Collection or the American Type Culture Collection in the US, or the Riken Bioresource Center in the Japan. So this is very few. This is actually, it has some consequence. And one of the consequences is that whenever you want to have fish cell lines, you have to directly request it to the developer, okay? We all know that most of the time they do not answer. It's very difficult to get a cell lines when you ask directly to the developer. And it's not only that, the fact that the cell line is available from a cell bank, it's also, how can I say in English? It's also a gauge of quality. Why? Because this cell bank actually will test the cells for the presence of contaminants, especially mycoplasma. They will also authenticate the cell lines. And it is very important because in the past, and it's not only something which is related to fish cell lines, it also happened in the other cell lines, there was some misannotation of some cell lines. And there is one example which is related to the EPC cell line, which was supposed to be originated from carp. And actually, after some time, and after some few papers published on that cell lines, they realized that it was from the minnow, from the fathead minnow, okay? So the fact that the cell lines are deposited in a cell bank, such as the ATCC, the ECACC, or the Riken, it's a quality parameters, which is very important. So just to mention that among the 11 cell lines that were developed in Portugal, is particular in the CCMAR, only one is available, and that one is available in the SCRCC. It's a cell line that was developed from the seabream, from the bronchial arch. And well, it's not enough, and we are certainly aiming at depositing more cell lines in the future in the database. Okay, maybe I will, I've already talked for 25 minutes, and maybe I should speed up a little bit, and maybe that one I will go very quickly on that one, because, well, it's a little bit boring. It's just to give you an idea of the different application for the fish cell lines. So as I mentioned before, historically, fish cell lines were applied for virology and immunology. So, and this is related to the many disease outbreak that occurs in the fish farm, okay? So it causes massive economic loss and impact negatively animal welfare. And then, so fish cell lines were developed to be used as diagnostic tools, research tools, and mostly they were used to host, propagate and detect the virus, but also to develop some vaccine and to study host pathogen interaction. So fish cell lines have been also applied, I mentioned that before, in ecotoxicology and toxicology, and mainly they are there to replace the experimentation that was done on whole animal, okay? So traditionally, fish has been used for ecotoxicology studies to test the toxicity of environmental pollutants, of water effluents, but it has also been used to test the toxicity of food additive, pharmaceuticals, cosmeticals. Okay, so the idea here to use the fish cell lines is to replace animal experimentation, and so it's used in place of the whole animal, in place of the fish. So biotechnological application, I will have few slides about that next, so I will go very quickly. So cell lines, and in particular embryonic stem cells, were used to produce transgenic fish. So why ES cells? Because actually they are easy to manipulate their genome. You can make them express a particular transgene. It can be a gene which is a resistance to a particular disease. It can be a gene that will improve the growth or will improve the reproduction. So ES cells, so embryonic stem cells, have been used and are still used, of course not in every country, because transgenic fish is not utilized in all countries. There's still some restriction. Some cells from fish are also used for cellular aquaculture. So cellular aquaculture is, I would say, cell-based seafood. It's to produce some food in a plate, basically. And you all have heard about that bovine steak that was produced in a plate in a lab. So there is now some companies, it's not that many, I can remember 2 or 3, that are betting on using fish cell lines to produce fish meat in a lab. Okay, so this is a promising field of research for fish cell lines, and I will talk a little bit more about that later. Application in functional genomics. So what is functional genomics? Functional genomics, actually, it's a set of molecular techniques that are used to assess gene, protein, or metabolite function, okay? And why fish cell lines are very important for functional genomics, it's because it's easy to, and especially now that with the development of the CRISPR-Cas9 methodology, it's very easy to genetically modify fish cell lines. And so you can have some studies that are aiming at gain of function or loss of functions. And actually fish cell lines, there is a number of articles that have been using fish cell lines, that have been using fish cell line for functional genomics. Okay. Let's go a little bit quicker on that. I will I would like to mention just the use of fish cell lines for species conservation. And actually, this is something which is not that much known, is that on the list of endangered species, the list which has been released by the EUCN, there is hundreds of fish that are endangered. And a way, well, the cell lines are actually a way to preserve the genetic material from these endangered species. So you would, for example, have some cell lines from the primordial germ cells, and you will just cryopreserve them until they are conditioned, for example, to recover these cells, and maybe using a surrogate reproduction to restart the production of a population of these endangered species. So this is something which is very new, and which is very important. So far, it exists some cells for some catfish that are endangered and for some sturgeon, okay? But there should be a huge effort to increase the provision of cell line from these endangered fish species. Okay, I was very quick on this, but if you want to read more about that, you can search in the future. It's still not published. It's a chapter that we have been produced for this book, which is edited by Ignacio Fernandez and George Fernandez. It's called Cellular Molecular Approaches in Fish Biology. And then we contributed with that specific chapter on the current status and future challenge of ray fin fish cell culture. You can also read this paper, which is an inventory of the fin fish cell lines that were developed from 2010 and 2020. But now, I've been speaking already for 30 minutes. I would like to speak quickly about what matters for the Bioscale Lab, which are fish bone derived cell lines. So why are we interested in fish cell lines that are derived from bone tissue? And it's mostly because fish have been increasingly used in the last decade to gain knowledge on the mechanisms underlying bone formation and mineralization. And among these fish, I would like to make a distinction between the small teleost, such as zebrafish and medaka, that has been used to model human skeletal disorder, such as osteoporosis, osteopenia, and to study the disease mechanisms of these, bone disease. And then I would like also to mention the fish that are used in aquaculture industry. And why? It's because these fish actually, they have a high incidence of skeletal deformities. And it's very important to develop some cell lines to be able to try to understand what are the cause of these skeletal deformities, okay? And that's what we have been doing in the Bioscale Lab, and I will talk about that soon. Okay, just to give you some historical data about fish bone cell culture. I told you before, there is a deficit in cell lines that have been derived from bone and that are capable of in vitro mineralization. I told you it's 12 cell lines out of 717 cell lines. So most of the cell lines that are available from fish are from soft tissue. And this is not a situation actually which is only related to fish. It's also for most of the vertebrate. And the reason for that, it's maybe because the diversity of soft tissue is higher than the diversity of hard tissue. You have much more soft tissue than hard tissue. But we think that mostly it's related to the difficulty of producing cell lines from hard tissue. And I explained that already in the first slides. It's much more difficult to get the cells out of a hard matrix than from a soft matrix. So it takes more time. The cells are usually more damaged. So we have less success in the development of fish cell line from hard tissue. So the first report of cell lines, fish cell lines derived from bone tissue date from 2004. And it was in our lab, in the BioScale lab. The cell lines, actually it was two cell lines. It was VSA13 and VSA16. They were developed from the gilded sea bream, Spirus and from vertebra. Yeah, it's not said there, but it was from vertebra. And actually, we developed these cell lines, and they have the capacity to mineralize their extracellular matrix in a matter of 3 to 4 weeks. Of course, you will have to provide them with a source of calcium, and in that case, we put calcium chloride, a source of phosphate, and we put beta-glycerophosphate, and we also put some ascorbic acid to improve the formation of the extracellular matrix because it's important for the maturation of the collagen fibers. So we have characterized these cells in terms of capacity of mineralizing the extracellular matrix, and we confirm that the crystals that are deposited in the extracellular matrix are similar to what we can find in fish bone. They are hydroxyapatite-like crystals. We have looked at different bone markers, either look at gene expression or enzymatic activity. And then we conclude that these two cell lines were probably osteochondral progenitor cells, okay? So this is just some examples, some picture of how we assess extracellular matrix mineralization in fish bone-derived cells. And here it's the example of the VSF13 cell lines. So as I told you, we will supplement the medium of the cells with a mineralogenic cocktail that contains calcium, phosphate, and vitamin C. And during the first week of exposure, the cells will differentiate. They will produce extracellular matrix that will have the ability to bind the calcium crystals. And after 2 weeks, you can already observe the mineralization of the extracellular matrix. And here you have some example. It's a macroscopic view of a multi-well plate containing VSF13 cells at 2 weeks and stained with alizarin red. Alizarin red is a specific dye. It's a calcium-specific dye, and it will react with the calcium phosphate crystals that are deposited in the extracellular matrix. So here it was a macroscopic view. Here you have more a microscopic view, and it's still the VSF13. It's still after 2 or 3 weeks of mineralization, but these times they have been stained with a different dye. It's Van Cossa staining, and that dye actually it's based on silver ions that will react with the phosphine, get oxidized, and give that particular black coloration. So if we use a scanning electron microscope to look at the extracellular matrix of the VSF13, we could see at different magnification the deposition of the mineral nodules. Here they are all identified with that green arrowhead. Okay, and I remind you that these mineral nodules are made of hydroxyapatite-like minerals. So if we look at the fish cell lines that are available to study in vitro mineralization, first we can see that they are all coming from 7 different fish species that are species that account for very important taxonomic group in Chondrostei, but also Teleostei. With 6 cell lines, the gilded sea bream is the major contributor of fish bone-derived cells. And the dominant position of the gilded sea bream versus, for example, the zebrafish, where there is only 1 cell line available from zebrafish, it's probably related to the activity of the BioScale lab. and to the fact that actually the gilded sea bream is an important aquaculture fish, and it has a high incidence of skeletal deformities in that species. So we wanted to understand, by developing these cell lines, what were the mechanisms of in vitro mineralization and maybe what could cause these skeletal deformities, okay? Another explanation for the fact that we have a lot of fish cell lines from gilded sea bream is that for some reason that we still do not understand, it's much easier to develop fish cell lines from marine fish, and gilded sea bream is a good example, but we have also developed from Atlantic salmon, from Senegalese soles. It's easier to develop cell lines from marine fish than from freshwater fish, okay? We don't know why. It could be related to the medium that we are using. I don't know if the medium that we are using are more suitable for marine fish cells than from freshwater fish cells. We don't have the clue about that. But our experience is that it's much easier to develop fish cell lines from marine fish, and in particular here, gilded sea bream. So if we look, something that is very important to mention, I didn't mention it before, is that these fish actually, the fish species that are that have been used to develop these fish cell lines have different bone type. So I didn't mention that before, and I will not talk extensively about that. But for us, which are working with bone, this is very important. You have some fish species that have a bone which is said cellular, so it has the presence of osteocytes. Osteocytes are cells that are very important for bone remodeling, okay? And you have some fish species, so for example, the sturgeon or the zebrafish are fish with cellular bone, and you have some species that do not have these osteocytes present in their bone, and they are called acellular bone, okay? So for us, it was important to develop fish cell lines from the two types of bone. So if we look at the tissue that were used to develop these bone-derived cell lines, most of them were developed from the vertebra, but also from bronchial arch, so vertebra, bronchial arch, from the jaw, okay, which is also a calcified tissue which is easy to sample. And in some cases, and in particular in the cases of the zebrafish, it was from a mixture of different bone structure. Why? Because of the size of the fish. It was very difficult for us to sample vertebra or to sample a particular calcified structure. So we decided to use all of them, okay? Maybe I should have also a particular mention for these two cell lines that were developed from sea bream. They were not developed from skeletal structure. They were not developed from vertebra, bronchial arch, or from the jaw. They were developed from, no, actually it's these two there, okay? They were developed from either the blastula stage embryo, and they were like embryonic stem cells, or they were developed from the regenerating fin, from the blastema, which is a tissue which is not pluripotent as the embryonic stem cells, but it has some multipotency, okay? What is important here to say is that these two cell lines, although they were not developed from skeletal tissues or from bone, from vertebra, they had the capacity, when they were treated with appropriate supplements, to differentiate into bone-forming cells, the osteoblasts, but also into bone-resorbing cells, the osteoclasts. So it proved their pluripotency or multipotency. So they had the possibility to develop into different bone cell type. Maybe to finish on that table, I would like to mention maybe the two cell lines that were developed from the Adriatic sturgeons. And some of you may know that in the Adriatic sturgeon, the cartilaginous elements are part of the non-calcified skeleton. And actually, these two cell lines, although they were developed from vertebra and from bronchial arch, but are in the other fish species capable of in vitro mineralization, these two cell lines were not capable of mineralizing their extracellular matrix. And actually, it's very important to understand the mechanisms that induced this mineralization. For example, if we compare the transcriptome, for example, of these two cell lines, one which is capable, they are both from vertebra, but one is capable of mineralization, the other is not. Maybe we could identify some genes that are critical for this in vitro mineralization. Well, maybe it's worth also to mention that all these cell types, cell lines, were developed from the bioscale line, except these two, which are from the blunt snoot bream and from the mosquito fish. Maybe a special mention to that from mosquito fish, because it was developed by Paramech, who used to be a postdoc in our lab. Finally, on that, maybe on that table, it's worth to mention that all these cell lines are adherent, form adherent monolayers, okay? They attach to the surface of the culture, and actually it's a monolayer, so they are contact inhibited, so they will not form multi-layers, okay? Well, let's go quickly on that one. It was a listing of the different application of the bone cell line. So we have applied them in toxicology to test the mineralogenic effect of some environmental pollutants. So some pesticides, some polycyclic aromatic hydrocarbon. We have used them in some pharmacological applications, so to test the effect of some hormones, some drugs on the mineralization of the extracellular matrix. Bioprospection, I will have some few slides about that just after. We have used the cell lines to screen some natural extracts for the presence of compounds that could have the potential to increase the mineralization of these cells. Okay, they have been used also for functional genomics to look at the transcriptional regulation of some important genes that are important for the mineralization of the cells, and to look at the anti-mineralogenic effects of some microRNA that are involved in the post-transcriptional regulation of some important bone genes. So I will now talk a little bit more about the use of these cell lines in bioprospection. So we are involved now for several years within the scope of several projects, some projects with people at the CCMAR, such as the Algared project, but also some people in Spain and in Portugal. It's the Blue Human project and the MAR 2020 project, the OsteoMAR project. So we are screening natural extracts. So it can be extract from seaweeds, from marine plants, from microalgae, etc. Also some terrestrial plants. And we have assembled a pipeline using different systems. All these systems are based on a different skeletal structure. So here we are looking at the developing operculum or at the developing vertebrae. We are also looking at the regeneration of some bone-like or bone structure, such as the scale or the caudal fin. We are looking also at some structure in the head. And we are using fish cell lines as a primary high throughput screening. Okay, so our pipeline is divided in two phases where we use low complexity bone system with a high throughput and high complexity bone system with a low throughput. So the aim is to test different extract, a fraction of this extract, and eventually to identify the compounds that have a bone anabolic effect or an osteogenic effect. And in this pipeline, the fish cell lines that we are using are mostly the VSF13, the cell lines that were developed for the vertebra of the gilded sea bream. This assay is very simple. So basically it's based on the spectrophotometric quantification of alizarin red staining. It's very quick, it's very simple. It doesn't need expensive material or expensive reagent. The only drawback of that system is probably the time that it takes. Oops, sorry, the time that it takes. So we need, and the VSF13 are kind of cell lines that are very quick to mineralize, but still we need 2 weeks to have the information if an extract is pro-osteogenic, anti-osteogenic, or if it has no effect on the mineralization of the extracellular matrix. Okay, so 2 weeks, and we try to compensate that by using multi-well plate. Okay, one good thing about that system also is that we use very small amount of each extract, and in some cases it's very critical because sometimes we have some collaborators that send us milligrams of product, of compounds, while we would need in some cases more grams. Okay, so these mineralogenic cell lines are part of the first step in the pipeline, so the screening of the extract or the more complex fraction. And what do we get? So after 2 weeks, we have, so this is picture of mineralized culture, and you have here the control, and then you have one extract that will be increasing the mineralization of the extracellular matrix, and so you will have a pro-mineralogenic extract, and you will have some extract that will decrease the mineralization of the extracellular matrix, and of course you will have some extract that have no effect. But what we are interested in is either in the extract that will increase the mineralization or in the extract that could decrease the mineralization. Okay, so here it's just, oops, there is already some annotation there. This is an example, and this is a slide that Joanna gave me. This is an example of extract that we received from Cyanobacteria. It was 7 different strains, and actually these extracts were tested first in the operculum of the zebrafish, and we identified, I don't know why, and we identified 14 fractions with different polarity that had a pro-osteogenic effect or an anti-osteogenic effect in that operculum system. So when we tested this extract in the cells, we got what? We got 7 extracts that had a pro-mineralogenic effect in the cells, okay? And it was also shown to have pro-osteogenic effect in the operculum. Interestingly, we got 2 extracts that were shown to have anti-osteogenic effect in the operculum, and you can see it here. You can see that here, I don't know why I cannot mark it. Yeah, you can mark it. You have here, you can see it. You have here the operculum of the control fish, and you have here the operculum of the fish treated with one of these antiogenic fraction. And actually, you can clearly see that the area of the operculum, the growth of the operculum was reduced, okay? And then when we test this extract in vitro, we could see that actually it could increase the mineralization of the cells. And actually, if you look very carefully into the operculum, you can see that this operculum is much more intense. The staining is much more intense than in that one. So it makes sense with the results that we got in vitro. So just to conclude on that is that the fish cell lines are very interesting tools to screen natural extract for compounds that could increase the mineralization of the extracellular matrix. And maybe these compounds, of course, it needs more testing, but these compounds could be the basis of future drugs for to treat skeletal disease in humans, such as osteoporosis, which is characterized by a low bone mineral density. So imagine that these compounds, which is there, which increase the mineralization of the cells, could also increase the mineralization of the osteoporotic bone, and maybe you could recover the defect of mineralization that you observed in this bone, osteoporotic bone. Okay, I was planning to talk about that, but I will not talk about this. This was a simple protocol just to show you how we develop the different fish cell lines. If you want to know more about that, you can look into the book chapter that I referred before, because it will be there. Just a small conclusion about the fish cell culture and what are the future perspectives. And I think the perspective is to really to increase the provision of fish cell lines for particular system. And I will mention the biomedical models. There are still very few cell lines from fish that are used as biomedical models for human disease. And very few cell lines, and also the tissue that are at the origin of these cell lines are not very diverse. So we should have an effort, we should make an effort by to increase the provision of in vitro cell system for this biomedical model. We should also maybe have an effort to increase the provision of cell lines for emerging and economically important aquaculture species. And here I could just mention the sardines, maybe the meager and the amberjack, the seriola, which are promising, which are emerging species for aquaculture. And it will be interesting to have in vitro cell system for these species. I already mentioned that, and this is something that is very important. It's to develop cell system for endangered fish species to help to preserve the genetic material from these species. We should, of course, increase the storage of fish cell lines in cell banks. I remember you that only 5% of the fish cell lines are present in the cell banks. It's not enough. We have to do better. I will not talk about that because I don't have time. It will take too much time. Just I talk about an effort in improving the provision of in vitro cell system from fish, but we should also make an effort in order to improve the methods. And here I mentioned the transfection. It is true that the fish cell lines, and in particular the bone cell lines, are very hard to transfect. Okay, so transfection is the delivery of DNA inside the cells. And this is a clear limitation, for example, for functional genomics, when you want to study the function of the genes. We have to make effort to try to improve these particular techniques. And then we have also to make maybe some efforts to develop specific reagent for fish cell lines, and specific in the sense that, for example, I mentioned to you the cell-based seafood aquaculture. It's very promising application for fish cell lines, but it's still lacking the development of serum-free medium for this particular application. And here I put an example of this company, Brunalu, that have successfully developed serum-free medium to grow the cells from, I think here it's the yellowtail, it's an amberjack. Okay, but this is one species, one company. There is a huge effort that should be done to optimize this serum-free medium for other fish species if we want to develop more this cell-based seafood aquaculture in the future. Okay, now just 4 slides and I will be done. It's just about what is happening in the CCMAR in relation to cell culture, but what is important for fish cell culture. So you may know, some of you may know, that the CCMAR has invested in a brand new cell culture facility. It has been financed by the EMBRC project. It will be located in the 3rd floor in the room 338. Okay, the room is built. Most of the big equipment have been delivered. We are just missing some small equipment and maybe the access system with the card. So then this room can be functional and can be used. And Well, it will be a normal cell culture facility. Actually, it will be a BSL2 plus facility. It will have a biological safety cabinet, CO2 incubator, inverted microscope, but importantly for fish cell culture, it will have 2 cell incubator which are refrigerated. That means that we could set the temperature. Of course, it's only 2 incubators, so we can only set 2 different temperatures, but we could, for example, set the temperature from a cold water species, the salmon, which is growing well at 20, 21 degrees, and the other one could be set at the temperature for a warm water species like the zebrafish at 28 degrees. But this is just example. But these 2 incubators will allow us to cultivate fish cells in the CCMAR cell culture facility. And the other, the last 2 slides is to let you know that the CCMAR is part of the recently funded collaborative lab, which is named S2 Aqua Collab. It's a consortium. It's led by the IPMA, and the CCMAR is one of the partners. I think it's about 15 or 16 partners. And the main goal of this collab is to foster aquaculture development. Okay, and then you may question what will be the role of fish cell lines in this collab, and it will be in the activity 3 of the work package 4. So it has not started yet because it has been just recently funded, but we aim in this activity to improve the provision of cell culture of cell lines from fish species that are important for the Portuguese aquaculture. And in particular, we would like to put some focus on that emerging species that I mentioned in a few slides ago, which are the sardine, the meager, and the umberjack, the seriola. But we will use also these cell lines to gain knowledge about toxicity of some compounds and also about some pathogens, so to look into host-pathogen interaction. Something that I would like to develop, but I don't know if we will have time and money to that, it's related to the biotechnological application of the fish cell lines, and in particular for cell-based seafood or cell-based aquaculture. So we would like to explore the possibility to use some of our cell lines or to develop some cell lines to see if we could develop in the lab some fish meat, or at least to explore the condition that could be applied to a future project, in a future project to develop fish meat. Okay, then the last slide is to thank the different people that have contributed to this presentation, or at least to the development of the fish mineralogenic cell lines, which are the former and present member of the Bioscale Lab, to the people that have helped me to collect data on the fish cell lines and write the book that will be soon published. So it's Joanna, Marco, and Leonor. The people that have been involved in the implementation of the CCMAR cell culture facility, João, Carla, Anna, and Adelino. And of course, the people, and I just mentioned here Katia and Pedro because I've been interacting mostly with them, but also all the colleagues from the collab. Thank you.
Thank you, Vincent, for really interesting and informative presentation.
Well, maybe a bit too long.
Yeah, but still. Anyone has questions? While people are thinking, I have 2 questions. The first one is when you talk about the functional genomics of using cell lines. Do you know how is the efficiency of a CRISPR technique on fish cells? Do you have an idea? Well, actually, it has been applied to several fish cells that were derived from several fish species. So, of course, it has been applied successfully to zebrafish, but also to other fish species.
So it's not a routine technique, that's for sure, but it's working. Okay.
Okay. And my second question is related to the new cell culture room here at CCMAR. There will be any possibility to storage more cells, for example, 1-150 ultra freezer. I don't know.
Well,
what are the ideas behind the room?
Well, it's a good question. Currently, there is not a cryogenic freezer available. The only one which is available, and actually you have two available, is from the CBMR. Okay, so if you go to the building 2, and that's actually where we have our cells, you have a cryogenic freezer there, but it's at the other side of the campus. So it would be nice to have a cryogenic freezer in the CCMAR, especially because there are more group working, not with fish cells, but there are more group working with mammalian cells, with other cells that currently, I don't know for all the group, but some of them are using liquid nitrogen. And actually liquid nitrogen, well, it's nice, it's working well, but it's dangerous. And in Algarve, it's not easy to get it. So really, we would need to have a cryogenic freezer. It was not into it was not bought within the scope of that EMBRC project, but hopefully it will be bought within the scope of another project. I agree with you. This is really I would say it's a critical equipment for the cell culture facility. Okay. Not yet, but hopefully in the future, because you would have a specific usage for that. You have a specific need for that. Why did you ask about that?
Just a curiosity. How do you gonna manage all those resources, and including on the collaborative laboratory? How you will storage the future cells that will be generated, for instance?
Well, maybe you're right. Maybe we could buy that within the scope of that collaborative lab if there is money for that, okay, or another project. I know that the Bioscale Lab is interesting into that, but I know also that there is 3 or 4 more group of the CCMAR that are interesting into that. So it will happen, I hope, in the future that we will have a cryogenic freezer.
It will be a plus for the center because you can allocate all the cells there, and it's
and then I mean, for the people that use liquid nitrogen, it will be safer, and it will be also more economic because it's very expensive to have liquid nitrogen, and you have to renew the liquid nitrogen not every week, but you have to ask for more liquid nitrogen kind of often. And for us, it will be interesting because then we don't have to go to the other side of the campus to get our cells.
Of course. Thank you. Anyone has other questions?
No.
Okay. So
well, anyway, as I told you, most of the data that I presented today, it will be in that chapter that we wrote about fish cell lines, and there is a particular focus in that chapter on bone cell lines. So if anybody wants to have more information, or if I was not clear, or I had to cut a lot of information, then I advise you to read that chapter. So they can ask it to me, and I can give them. Okay. Just to
and all this information you are about to publish in a book?
Yeah, that's what I mentioned there. So most of that information, the table, the figures, will be, and all the analysis of the data from the Celosaurus will be published in that book, which is from Ignacio and George. It's about cellular molecular approach in fish biology, and there will be a specific chapter on fish cell lines. Okay, it will be available very soon. In the meantime, if people want to read more about fish cell lines, they can look at that paper, that recent paper from Tangeray. That I think it was published in 2021. Yes.
Thank you for the really nice talk, and informative talk. So see you next week. with the following CCMAR seminar. Thank you all.